Hello, this is the third part in the lecture series Food Microbiology. In the next couple of lectures, we are going to see how do we detect the presence of microbes in the food, number 1. 2, if microbes are present, what is the quantification methodology? And 3, whether the food is safe to be consumed. 4, identify the microbes. Is it pathogen or is it a spoilage organism? When there are microbes present in the food to be consumed, food spoilage can be of two ways. Number 1, the growth and active metabolism of food components by the live cells, meaning when the microbes proliferate in the food particle concerned, they will utilize the nutrients present in the food. As a result, so their metabolism, active metabolism will absorb the nutrients. So slowly the food will get spoiled. That is the first case. In the second case, even in the absence of live cells, by their extracellular and intracellular enzymes that react with the food components and change their functional properties, leading to spoilage. That is, even if there is no live organism present in the food, if the bacterial enzymes are present, those enzymes can react with the food components, changing the components' nutrient properties and the functional properties, thereby leading to the degradation of the foodstuff. To reduce the loss of raw and partially processed food by microbial spoilage, we consider 2 points to be very important. First is, what will be the expected shelf life under normal conditions of handling and storage, meaning without using any precautions, if you just normally package the food and store it under normal conditions, how long can this food be consumed, like safely, without it getting spoiled, without it giving any food poisoning or any other negative issues. The third is to determine the current status with respect to spoilage of a food that has been stored for some time. And after some time, after you have stored it, say for a couple of days, you can take it back and assess what sort of a damage has happened, what is the sort of microbe which is growing there, what is the type of spoilage which is happening to the particular food particle concerned. Many criteria have been evaluated for their efficiency as indicators to predict the expected shelf life as well as to estimate stages of microbial food spoilage. Now, this means that when the food has been first processed and kept, like either direct as a fresh food or as a freshly processed food, what would be the nutrient aspect, the flavor aspect, what would be the things you look for in that particular aspect. That is number 1. 2, the same food as it is kept on the shelf for n number of days without getting spoiled, what have been the changes happening to these qualities which you consider to be important. And 3, at the end of the shelf life, what you deem, what of the sort of the microbes which have come up and what has been the degradation pathway which is happening in the particular food concerned. So these criteria or indicators can be grouped as 1, sensory parameters, microbiological parameters and chemical parameters. So these are the main indicators to predict the expected shelf life as well as the damage which has happened to the particular food concerned. The sensory tests, which means the changes in the color, odor, flavor, texture and general appearance. Changes in the texture and flavor generally appear in the advanced stage of spoilage, that is, as you have microbes growing and proliferating, because of their proliferation, because of the changes which their enzymes cause to the composition of the food, you will have changes in the flavor and the texture as well as odor. So that means that the food has already started getting spoiled. Odor changes can be masked by spices which are used in many products. So odor needn't be decipherable. You needn't actually find out that there is an off odor if it is very less. Finally, individuals differ greatly in the perception of the organoleptic criteria. Some people can make out even if there is a slight change. The sensory evaluation by such people would be very critical, whereas some people can make out that there is a deterioration only when the change has been drastic. So individuals will differ according to this perception. The contributing factors in microbial spoilage of food include the type of product, its composition, the methods used during processing, contamination during processing, the nature of the packaging, temperature, time of storage and possible temperature approach, which means that as we have been discussing the type of the food, plant or animal product, how it is water activity and the other things which make it more conducive for the microbial growth, its nutritional composition, then what have been the conditions during processing, how has it been packaged, then what has been the hygiene level during packaging, then the temperature of storage, and finally, has there been drastic change in the temperature during storage. So, all these factors influence whether the food is going to be spoiled or not. So, what are the baseline indicators? In a good fresh product, it can be present as a low number, that is microbial, or absent in chemical parameters. Under normal conditions of storage, that is temperature, time and packaging, it should increase, that is the microbial level or the chemical level in increase in quantity to reach a very high level. And when spoilage occurs under normal storage conditions, it should be the predominant causative agent. The agent of damage, whether it is chemical or microbial, it has to be detected very rapidly. Then it can also be used reliably to predict the shelf life and the spoilage status, meaning if you detect this much level, yes, this indicates that the food is spoiled. This will predict that yes, the food has been spoiled, or no, the food is safe to be consumed. And it should have a good relationship with the sensory criteria of spoilage of a particular product. So, whatever is the indicator we are taking, whether it is microbial or chemical, should fulfill all these baseline conditions for it to act as a baseline indicator. Normally, for a microbial spoilage, the tests we do are: one, the standard plate counts, otherwise called as the SPC for the viable cells; the most probable number, otherwise called as the MPN method, as a statistical determination of the viable cells; the Rye reduction test to estimate the numbers of the viable cells that possess reducing capacities; direct microscopic counts or DMC for both viable and non-viable cells. So, these are the standard tests used in microbiology, not only for food but the evaluation of microorganisms. In food also the same things we use. SPC is the most widely used method for determining the numbers of the viable count, or in other words we call it as a colony forming unit, CFU, in a food product. So, what we take into concern if each single organism which we detect will form a single colony. So, if you take a sample, suppose you scrape off a sample from the food, dissolve it, suspend it in a medium and then plate it on a agar plate. If you see 100 colonies, it means that each of these 100 colony, that single colony is the progeny of one bacterium. So, you had 100 microbes in that particular sample, or in whatever was the aliquot, depending upon the dilution you took. So, each single organism forms one colony. So, this is called as a viable colony forming unit. This in turn, to count or to enumerate the number of the CFUs, depends upon what is the sampling method which we use, and also the distribution of the organisms in the food sample. Third, the nature of the food biota, nature of the food material, the pre-examination history of the food product, nutritional adequacy of the plating medium employed, incubation temperature and time used. Now, sampling methods, like from a mass of, say for example, if I have a 10 gram of the sample, how much did I use, what was the sampling, how many random samples did I take, that is number one. 2, in that particular food sample, what is the distribution of the microbe you are looking for. Remember, in the earlier lectures I said that normally pathogenic bacteria, they occur in very less number when compared to the normal flora. So, now the question is, are you looking for identification pathogens, or are you looking for the identification of normal microbiota, which are spoilage organisms? So, the methodology for each is going to be different. The nature of the food biota, what is, depending upon the product, whether it is a plant, animal or a marine, seafood, the nature of the microbiota on that particular food will vary. So, accordingly we will have to change the concentration and the type of sampling method which you are going to use. Nature of the food material, is it processed, is it fresh, what is the condition of the particular food product, what has been its storage conditions, all those will influence that. Then pre-examination history of the product, meaning have you taken into consideration, suppose it was a plant, was the plant, did it have any microbial attack or a fungal attack when it was in the field, what was the conditions, what were the precautions taken by the farmer. Or if it is an animal product, for example, you are talking about meat, what was the health status of the animal, has it been fed with antibiotics, what was the hygiene level in the place where it grew up. So all those pre-examination. Then when you take the sample, its condition, what was the nature, so you have to observe all those. Was it neatly packed, was the pack, did it have any breakage, what was the condition, what was the temperature. All those things will also determine the microbiota which are present on the particular sample concerned. The nutritional adequacy of the plating medium. What medium have you chosen for the microbes to grow? It should contain the carbon, electron and the energy source for the growth of the normal microbiota or the microbiota, specific microbiota concerned. So that nutritional adequacy. Then finally incubation temperature and the time. Here you can vary depending upon whether you are going to trying to find out whether it is a macrophilic organism or a thermophilic organism. Accordingly, the plates can be kept for incubation in different temperatures for different times, and accordingly you can find out what is the category of the microbe which has grown up in this particular sample which you have taken. pH, water activity and the oxidation reduction potential of the medium, which in turn will influence the growth of the microbes. The type of the diluent used to dilute the microbial load which you have scraped off from the sample concerned, and finally the relative number of the organisms which is present in the food sample. So what grows up in a plate in turn depends upon all these factors which you have identified in the food sample concerned. Indicator organisms. To identify the cause of spoilage or the presence of pathogen where a food has been implicated in food illness. So here we are going to try to find out a pathogen in a food sample. Normally itself, among the microbial load, the pathogen load in a food is going to be very less. So instead of trying to find out the low number of the pathogen, if you have another organism which normally grows along with the pathogen but normally grows in a higher number, so it means that the pathogen wherever the pathogen grows, this organism will grow, but pathogen the growth would be less or it will be lesser in number, whereas this organism would be present in a large number. So such organisms can be used as indicator organisms. In other words, you say that if this many number of the indicator organism is present, automatically the food sample might also contain the pathogen concerned. So the method used for determining an estimate of the total mesophilic count, it is very different from those which are required for demonstrating the presence of a pathogen. The isolation of specific pathogens which may be present in very low but significant numbers in the presence of larger numbers of other organisms requires elaborate procedure. So for this isolation of pathogen, we require enrichment in the media which will encourage the growth of the pathogen while repressing the growth of accompanying flora, meaning from the mass of the normal microbiota which grows up in a selection medium, we have to enrich the medium in such a way that only the smaller number of pathogens grow up, whereas the other organisms are inhibited or growth rate becomes low down. Then you have to isolate on a selective diagnostic media, meaning once such putative pathogen samples have grown up in the medium, you now have to confirm that the organism which has grown up is the pathogen. So most of the cases the pathogens have a specific media which would be specific for them alone. So these colonies which you have isolated in your plate, if it transferred and it grows in the selective plate containing the medium specific for that organism and the organism grows there, 100% sure your sample contained that particular pathogen. Then finally application of the confirmatory test. These confirmatory tests normally would be the biochemical tests which confirm that yes, this organism which has been identified has been 100% certified that it is this pathogen, not anything else. So you enrich the media, then plate it, isolate it on a selective diagnostic media and confirm it by the biochemical test. That is one method. The second alternative is to monitor for specific pathogens, is to look for an unassociated organism present in much larger number. So this was what I said earlier. Along with the pathogen, you will have some other organisms which proliferate in a larger numbers. So wherever you see the larger amount of this non-pathogenic organism, along with it you will see the smaller amount of the pathogen. So this is a concept which has been developed originally for pathogens spread by the fecal-oral route in water and which has been applied to fruits. So normally portability of drinking water is tested. Normally we look for the enteric pathogens, gram-negative enteric pathogens. There the indicator normally is E. coli. Yes, E. coli is a normal human gut microbiome, gut dweller. We accept a specific level, but in the drinking water, if the amount of this E. coli and the gram-negative organisms are beyond a particular level, then the potability of the water becomes a question. So that is a typical example. So then we say that this drinking water has been mixed with sewage, or there is some contamination. This concept actually was developed originally for pathogens which are spread by the fecal-oral route, especially in water, and now we apply it to foodstuffs also. How do we assess that an organism is a good indicator? Number one, it should always be present when the pathogen is present. Two, it should be present in relatively large numbers to facilitate its detection. Not only larger number, but that larger number should facilitate easy detection of the particular organism concerned. It should not proliferate in the environment being monitored. Its survival should be similar to that of the pathogen for which it is going to be used as an indicator. E. coli is a natural component of the human gut flora, and its presence in the environment or in foods generally implies that there is some history of contamination of fecal origin. In water microbiology in temperature climates, E. coli meets this criteria very well and has been proved to be a useful indicator organism of fecal pollution of water used for drinking or in the preparation of food. So quality of drinking water, as I said, is normally assessed by looking at how many CFUs of E. coli you get when you plate it. You just take a sample, dilute it, take a sample of water, you have your plating medium, plate the water concerned in the plating medium in appropriate dilutions and incubate it. Then look for the colonies which grow up in each dilution. So depending upon the number of colonies, you can assess whether water sample which you took, whether the amount of E. coli present in it is normal or it is higher than normal. If it is higher than normal, we indicate that it indicates that there is a mixture, that it is contaminated. So this is a normal method by which we assess the quality of drinking water, which we now can take as an assessment of food, because for washing, for the preparation of the food, if the water which we use has been contaminated, this will give an indicator. This will act as an indicator. For each of the food stuff which we use, we need to talk about this methodology. Number one, refrigerated raw or fresh meats stored anaerobically. So here, for assessing the quality of this, what we look for is enumeration of the CFU. That is, you look for the number of colony forming units per centimeter square of the psychrotropic aerobes, especially the gram-negative aerobes. So how many gram-negative psychrotropic aerobes are growing per centimeter square of the meat sample concerned? That is one. So the data can be available in 2 to 7 days depending on the agar media and the indicator, the plating methods and the incubation temperature, that is 10 degrees to 25 degrees centigrade used. So this will indicate the contamination level. When the same refrigerated raw or fresh meat is stored anaerobically, vacuum packaged, here again we are going to look for the presence of psychrotropic lactic acid bacteria. We use a suitable plating medium, agar medium adjusted to pH 5, because we are going to look for a lactic acid bacteria, as well as psychrotropic enterobacteria. Especially these enterobacteria here, the selective medium for their growth is violet red bile glucose agar medium. Along with it, we can look for the growth of some gram-negative organisms also. So what we are trying to see is, for depending upon the food product which we are talking about, what is the sort of contamination we are looking for and how do we test, because the type of microbiota which grows is different. So accordingly, you don't have to do all the tests. You need to look for specific microbiota which grow on those organisms, then look for their level. So for that we choose this. If it is raw milk, we go a species, we look for the cyclotrophic gram-negative bacteria as well as the thermoduric bacteria, which can sustain, which can grow beyond the pasteurization temperatures, which can still spoil the milk concerned. In pasteurized milk, again we do the SPC, we look for the presence of cyclotrophic bacteria, both gram-negative and gram-positive. In butter, we look for the lipolytic microorganisms which can cause rancidity, which will break down the fatty acids. In cottage cheese, again we look for cyclotrophic organisms, especially the gram-negative bacteria. So, depending upon the type of food which we are talking about, the type of microbe whose number we are trying to elucidate and enumerate will vary. In fishery products, we look for cyclotrophic gram-negative bacteria. In beverages, we look for aciduric bacteria, yeasts and molds. In salad dressing and mayonnaise, we look for lactobacillus and yeast. The major disadvantage of the microbiological enumeration method is that it takes, number one, several days for the population level of the indicator microorganisms from enumeration of COS to become available. So, as we saw, you will have to incubate it. For example, for yeast and mold, you take 3 to 5 days, and depending upon the plating medium also it will vary. So, this is a negative disadvantage, especially if you want to detect pathogen, because then you cannot send out, or sometimes you may have to recall what has already been sent out to the market. So, in such a cases we will require rapid methods. So, those rapid methods we will see later. Now, let us see what is the sample and the sampling plan, because this is the first step in your diagnostic tests. The microbiological quality of a batch or a lot of food, which could be liquid, solid or semi-solid, is determined by testing a small portion of a total amount. So, whether it is a liquid food, a solid food or a semi-solid form, what you take, the 1 gram or 100 ml, is going to be a sample. So, whatever you get from this sample is now the values you observe is now going to be implicated to the whole lot which you are going to be talking about. And this portion, which is called as a sample, and it must be a true representative of the total mass. Development and implementation of effective sampling plan is particularly important for the highly virulent pathogens such as Salmonella or E. coli 0157:H7 or a potent toxin as botulinum. So, even if you take 1 milligram, so 100 milligram of a sample and assess, and in that you suspect you identify the presence of Salmonella or E. coli, which are extremely virulent, it means that the whole food has to be dumped. It is a waste, it is not fit to be consumed. So, whatever is the plan which you are implementing for your experimentation, that development and implementation is very, very important, especially when we are talking about the extremely virulent food pathogens. Statistically, we have several sampling methods, of which I will mention a few. The first is what we call as a single attribute plan. Here, a lot is sampled once and tested for the bacterium or the bacteria of interest. The results are used to determine whether the lot should be rejected or accepted on the presence or absence of E. coli, for example, 0157:H7. The sample size or the number of samples N to be tested is dependent on the batch or the lot size. Defective units of N are dependent on the microbial type. So, you do the testing only once and you test it once for the organism. Now, depending whether you get a positive or a negative answer, then you will decide whether the whole lot has to be can be confirmed or it can be rejected or accepted. Now, and the sample size, it can be random samples taken from a series of lot. Each batch, you can say I will take 2 from this batch, 2 from that batch. So, that depends upon the batch size on the lot size. Multiple attribute plan. This is initially based on the lot size. The number of samples, which is the N1, are tested. So, if the defective units are above the acceptance level, so you form a baseline level here, and then you find out whether your experimental data is equal to this above the acceptance level or below the acceptance level. If it is above the acceptance level, you do a second sampling. So, like that, and then you analyze for defective units to decide whether to accept or reject. So, you already have a baseline of whether yes, if it is below this I can accept, if it is above this I will do a second testing or a third testing. Then if in all these it is above, then I will go and reject the particular sample concerned. The 3-class attribute plan. Here the microbial levels are divided into 3 groups. The less than or equal to M is acceptable, greater than M but less than or equal to capital M is marginal, and if it is greater than capital M it is unacceptable. Okay, so we have already fixed a value. Less than this I will accept, less than this or equal I will accept. Then I will put a threshold level which is in the upper margin. Anything between this small m and the capital M I will accept, but if it is beyond the value of what I have attributed as capital M I will reject it. So the number of units allowed between greater than small m and less than or equal to capital M among the samples tested, the small n is defined as for a particular microbial type. So for each microbe I will fix these values, and then according to the fixed values I will either accept or reject the particular food concerned. Plan level for low level of contamination. This is important for some highly potent pathogens. Units depending upon the lot size are tested to determine the presence or absence of the pathogen to make a decision. So even if total absence of pathogen you can accept, but even if there is a low level presence, very low level presence of the pathogen, then you really have to find out whether the food has to be consumed, whether you can accept it or whether you can reject it. So low level, what is the level? The individual collecting the samples should know why the samples are being collected. Now we come to who is doing the experiment. How there is a methodology. So once you have your sampling plan, the individual who is collecting the sample has to know why the samples are getting collected, what will be the type of testing which is going to be conducted, whether you are going to look for APC or are you going to look for coliforms or specific pathogens. Then the person should also have information about the product, whether it is going to cause food outbreak, foodborne illness, the nature of the product, etc. Then you identify the sample, label, nature of the sample, what is the type of analysis which we concerned. Then once the sample is collected, it gets transported to the laboratory under conditions which will avoid microbial contamination. And finally, in the lab, as soon as the sample is received, the person who receives it will note all these conditions and start processing the samples immediately. And if the sample has been damaged during transport, that has to be noted and rejected. So in this particular lecture, we saw how the importance of assessing the microbial load in a particular sample, how do we test for the pathogen which is present, what are the baseline indicators, and what are the steps taken, like starting from sampling to what is the care taken for to transport the sampling media and the food particle to the laboratory concerned. In the next couple of lectures, we will see the different types of quantification methods. Thank you.